I'm just going to give it another minute, folks, and then I'll introduce Tiara. All right, so thanks for coming, everyone. I am super excited. This is the last seminar of the year. Is that right, Paul? I think it is. Seeing yes. So I'm super excited to be introducing to you today Dr. Tiara Moore, who is currently in Seattle, but will be driving to Atlanta on Monday. So she might be a little bit distracted. But let me give you a little bit of background information on Tiara, and I'll pass the floor on to her. So Tiara is originally from Greenwood, South Carolina, and she did her bachelor's in science. She completed that in 2011 at Winthrop University, which was followed in 2013 by a master's in science from Hampton University. This is in Virginia. And then she went all the way to Los Angeles, where she completed her PhD in 2019 at the University of California in Los Angeles, so UCLA. And now she is a postdoctoral fellow working with the Nature Conservancy in Seattle until Monday. And she uses eDNA to assess ecosystem biodiversity. This is the current work that she does. I want to mention a few more things about Tiara. So I actually met Tiara when she was stepping down as student representative of ASLO. What is ASLO, you say? ASLO is a mouthful. It is the Association for the Sciences of Limnology and Oceanography. So in essence, it's a society that puts together, or that groups together folks that work on the marine world and the freshwater world, lakes, rivers, estuaries, and oceans. And she was on the board while I was coming in as deputy editor-in-chief of their flagship journal, Limnology and Oceanography. Now we have both moved on, but she was just such an important fixture of the ASLO board. And she wrote a really powerful piece, which I like to discuss with my students often. And maybe I'll drop a link in the chat at the end of her talk. But in essence, the title of her piece is "The Only Black Person in the Room." And so she does also, along those lines, she does a lot. She's a powerful voice and advocate for increasing inclusivity and representation of visible minorities in science, particularly in marine science. And I can just she has a long list on her CV, so I'm not gonna let them all. It's amazing that you're still productive with all that you do, but she is the founder of something called Black in Marine Science, and she is a powerful voice, especially for women of color in the sciences. And on that note, I will pass on. Thank you so much, Tiara. I mean, one of the wonderful things about the pandemic is that we get to see you, even if it's over Zoom, and we get to hear about your research. And so I'm passing the floor on to you, and I'll go into hiding until it's time for questions. Thanks, Tiara.
Thank you so much, Maggie, for that great introduction. When I give talks and people introduce me, it's just like, what? They're talking about me. So thank you so much for that. And thanks so much for the invitation and for allowing me into your space wherever you are right now. As Maggie mentioned, I am going to be moving. So I'm standing right now because I'm selling off my furniture. So my apartment is a bit of a mess behind me. So please excuse it. But I do want to just start my talk by acknowledging the stewardship and honoring the Duwamish and Coast Salish people on whose land I reside. And so the title of my talk today is Environmental DNA and Biodiversity Assessments, Establishing Tools for the Restoration in the Anthropocene. And so, let's get these slides going, okay. And so hopefully I'm going to talk to you today about how I use environmental DNA and these biodiversity assessments as a tool to really try to understand and figure out the impacts of climate change in the Anthropocene and hopefully lead to ways that we can mitigate and really tackle these issues for these impacts. And then, as Maggie mentioned, I also do some other work in the field, and I'll talk about that later. But I like to begin all of my talks doing my favorite thing, talking about me, just to give a little bit of perspective. And then, of course, I'm talking to students, so I like to be transparent about my journey. And Maggie did mention a little bit of this. So yeah, I'm originally from Greenwood, South Carolina, if you're wondering about my accent. And then I actually went to Winthrop University to be pre-med. Actually, I thought I wanted to be a pediatrician. And I realized quickly that I didn't really like kids like that. And so I needed to figure something else out to do. And so I started taking other biology courses, and I found a class called tropical ecology. And I'll be honest, the only reason I signed up for it is because they were going to Costa Rica for spring break for a research experience. I was like, oh, free trip. But when I got there, we were on a boat and we were collecting water samples and we were doing experiments, and there were senior scientists there. And I was like, wow, is this a real job? Is this a career? And I found out about the whole world of marine science. So I came back, applied to master's programs. And I put in my personal statement. This is important. They do read your personal statements. Hey, I took this one class. I'm very excited about marine science. Please accept me. And they did. And so I ended up going to Hampton University in Virginia, which is a historically Black college and university. And that's where I say I became a scientist and did research on the water quality in the Chesapeake Bay. Then, as a true scientist, I needed to become more educated. So I went on to continue to get my PhD at UCLA and continued work in the estuary down there. And I'll talk about that research a bit. And now I have this cool position, a partnership with the University of Washington and the Nature Conservancy, using environmental DNA to truly try to understand the changing environment. And I'll talk more about that research. But ultimately, my research and the things that I look at in my career is humans, human impacts. Humans are causing major environmental concerns. We know through history that because of the increased population, there is just so many different changes in our environment. Things like destruction of habitat, because again, because of the population, we don't have enough Walmarts and Targets, so we destroy entire ecosystems in order to build. Over harvesting, and the things like overfishing, because again, we have a lot of folks that we need to feed, so we mass fish and mass pull these organisms out of their ecosystems, and that can have damaging effects. And I'm not saying everyone should go and be a vegetarian or vegan, but I am saying that we do need to think about how we're taking and removing these organisms from their ecosystems, because they are causing problems like loss of biodiversity. So you can imagine if you destroy an organism's ecosystem, or if you remove it from its ecosystem, then the biodiversity of that ecosystem would then change. And that's impactful on the ecosystem and the organisms in general. Another major human impact that I've become interested in is nutrient enrichment. And so again, because we have large agricultural practices and things like that, we spray these crops with lots of fertilizer. Then as soon as it rains or a water event occurs, all of those nutrients are washed into the waterways and different water bodies. And this can lead to things like eutrophication, which is the buildup of this organic matter. And we can see this in the form of phytoplankton blooms, but here you see a macroalgal bloom. Another major human impact that we hear a lot about today is climate change. And this is, of course, this is due to the buildup of greenhouse gases like carbon dioxide in the atmosphere. And this is from, increases in transportation, large manufacturing, production, all these things that are occurring in the atmosphere. And actually found this little cartoon that I thought was funny. And here's the planet, the Earth, at the doctor. And it's like, oh, wow, your results are in. It's climate change. How many greenhouse gases have you been consuming? And so we see climate change is occurring because of these greenhouse gases. And I guess the most active, or the one we hear about the most, is carbon dioxide. And when we think about carbon dioxide, we know they can also lead to ocean acidification because carbon dioxide is absorbed by the surface ocean. There is a chemical reaction with the water, and that causes the pH to decrease, causes the water to become more acidic. And so that's harmful for our shelled organisms. So here you're looking at an intact shell that's just there. And then it's been subjected to low pH levels, acidic levels that are expected by 2021. And so you'll see there by the end, the shell is basically dissolved. So this is the equivalent, of someone coming into your apartment right now and just taking your walls down right around you. They're losing their protection that they need in order to survive. Hold on, something's going on with my slides. Okay. Okay. So right around the time when I was like trying to figure out what I wanted to do research on and trying to understand different changes in the ecosystem, I was really interested in this nutrient enrichment and these algal blooms that we were seeing, but then also this potential for ocean acidification. I was really interested in those two, I guess, concepts. And so I read a paper that actually talked about this phenomenon of like estuarine acidification or this eutrophication driven acidification. And so I'll explain this to you. I'll go through it, I promise, step by step. So if we think about this idea, right? So we have that land runoff, the nutrients that I've mentioned that come from, fertilizers and things like that, gets into the water column, right? And so that could lead to a macroalgal bloom, like I mentioned. So if we look at the productivity of the bloom, we know photosynthesis is occurring, right? So that is the production of oxygen and the consumption of carbon dioxide. But what's really important about this relationship and this whole, I guess, concept is that the macroalgal bloom doesn't stay intact, right? It will eventually start to decay and sink through the water column. And during that time, we would see the opposite of photosynthesis. We would see respiration, increase in microbial respiration, right? So the byproducts of respiration then are the consumption of oxygen and the production of carbon dioxide. So here, is that carbon dioxide that I talked about that's in the atmosphere that could cause acidification. But here is a local input of this carbon dioxide due to the respiration of the bloom that could also potentially lead to acidification. And then since I was working in a very shallow estuary in Southern California, I also included the sediment because the water column and sediment are tightly linked. So that same organic matter would eventually end up buried in the sediment. We could again see that increased microbial respiration, the production of carbon dioxide, and acidification. Also, interestingly, in the sediment, we know that things are buried and also recycled. So we could potentially see the recycling of the same nutrients that caused the bloom in the first place flux back out into the water column to then start a whole new bloom. So this was my idea. This is like, okay, if we can have acidification from carbon dioxide here, we have a bloom, that's the direct input of carbon dioxide, maybe we'll get acidification. This was to be my life's work. So I took myself out into the estuary as a marine ecologist. I collected, here I'm sitting in the midst of a bloom in an estuary in Southern California, have a pH meter, some buckets for collecting. I was taking nutrient samples and pH measurements. And so I found in the areas where we did see the bloom, these bloom decomposition, that it actually was acidic. In the areas where we didn't have the bloom, where we didn't see this same eutrophication, there was no acidification. So for me, yay, check. Theory proven. But as scientists, we know we talk to folks about our work, and someone mentioned to me like, oh, wow, this is really cool. But it would be interesting if we could see or understand who the microbes were in the ecosystem, who the players were that were contributing to this respiration. So again, as a marine ecologist, I'm like, what? How we go about doing something like that? I collected these measurements. And so this is when I found out about the whole world of environmental DNA. And so I like to explain this as, because everyone loves crime shows, right? And so, there is a criminal who does something and then they're leaving out and they leave a fingerprint. And then the detectives come in like, oh, the fingerprint. And they are able to get the DNA and they can tell you who that criminal is. And so basically, that's what I like to say I do in the ecosystem, because in the environment. So as we're moving, as animals are moving, as folks are moving through the ecosystem, we're shedding skin, fur, leaves, pollen, all of that has genetic information in it. And so we can potentially collect that genetic information with a soil, water, or sediment sample because it's just there. So if we look at this gift that I have, and I'll blow it up, right? So we see this fish that's swimming in the water. And so it's shedding DNA as it's moving through the water. So I could come in, scoop up that water, extract the DNA, and tell you that that fish was there. So this is how it would look. Oh, I always forget I have this slide and then I say that whole explanation. So you would collect the sample, then extract the DNA, then you could choose barcoding or meta barcoding, depending on what you're looking for. And then that could identify the species there. So this was really cool and new to me at the end of my PhD, that I was finding out. I was like, oh, wow, can I do this? And so I was able to run some of my samples in comparison where we saw the acidification and where we didn't. And when we looked at the biodiversity of the microbial community, just without having a background in microbial ecology, we could see immediately a difference in the middle site. In just the colors, looking at the different colors, and you're looking at the relative abundance of the microbes. And so the middle site is where we saw the bloom. And so when we look more into the biodiversity and the construction of this, we found that the organisms that were most present were decomposers and so we know that they were decomposing, so those were in the most abundance. And so I mentioned that the other bacteria that was present in both the mouth and the head are these Flavobacteriaceae, or just who are just common marine sediment microbes. So it was good to see those as well. So it's like, okay, we see this common, but we saw the overwhelming abundance of these decomposers in the middle site. So that was a link like, oh, wow, we did see this acidification, but we also saw the players that were present. And so that was cool to add to my PhD. And so I went to present this work. And I say this because, again, talking to students and talking about the importance of talking about your research. And so I went on, if you have the opportunity to go to conferences, definitely do it. And I'll also say this, as a student, there's usually student travel grants. So you shouldn't necessarily have to pay for conferences. So I know that can be a barrier sometimes, but there is opportunities for you to go to conferences. But anyway, so I went, presenting this research, basically similar to a talk that I gave you all. And someone came up to me, and this is why this is important. Someone came up to me afterwards and was asking me about eDNA and biodiversity assessments and how we can understand just changing ecosystems. And so I was just, explaining it to them, trying to answer the questions, as nervous as I was answering questions to a senior scientist after this talk. And then he asked me, well, could you do this type of research? Could you understand research of soil biodiversity in a forest? And so, I'm thinking about it. I'm like, well, yeah, sure. The methods will be similar. We would need to do this or this to figure it out. And he asked me, well, could you do it? And I said, in a forest? I'm a marine scientist. Like, what do you mean? And then he asked me one of the most profound questions I've been asked in the world. And he looked me straight in the face and he said, well, dirt is dirt, right? And of course it's not. But I make this point to say, as a student, we can think that we're like pigeonholed or we're tied to a certain ecosystem. But what he was telling me is that I had a transferable skill that I could potentially utilize in a different ecosystem to answer similar questions. And so as someone who was at the end of the PhD looking for a postdoc position, this was really intriguing that they wanted to do this project. And so that is how I ended up at the Nature Conservancy working in a forest, even though my heart is in the estuary. So I'm going to tell you about my next project quickly. So the Ellsworth Creek project, the Nature Conservancy purchased, basically there was this 8,000 acre old growth forest that was clear cut, basically for reasons that I mentioned, for logging, to make paper and things like that. And so the Nature Conservancy, which is the conservation organization, purchased it in order to restore it. And the whole goal was for them to replant the trees that we lost. And I just put this clip of this article that we had in the Seattle Times about this project, just showing the difference of the um, university or academia versus nonprofit. It's more like outward facing in science communication. So I do a lot more of that. But to really, understand why an organization like the Nature Conservancy, right, would even purchase a whole forest in order to restore it, we need to understand the importance of trees. And so when we look at trees, we know that they provide habitat, nutrients, resources, food, but most importantly, carbon sequestration. So when we look at the carbon sequestration of trees, trees on average can store 88 pounds of carbon. And so, of course, that's dependent on the size of the tree, the types of trees, different things like that, but on average, 88 pounds. But what's interesting though is that humans are emitting 44,000 pounds of carbon per year. So that's a whole lot of trees we would have to plant in order to offset that. So when we think about trees and the types of trees and the things that we want to have the best trees, we really have to understand the importance of soil. And so we know that soil also provides habitat and nutrients and resources. It also is the place really where trees live. And so when we understand soil, we also understand trees. So to the naked eye, right, you're looking at two trees. If I was just walking through my neighborhood, I'd see two trees. But when you understand the soil conditions, you see healthy soil, right? You see a more plush, vibrant tree that will be able to sequester more carbon. Over here on the unhealthy soil side, yes, you have a tree, sure, but it's not as plush, it's not as bright, it's not as productive, it's not going to sequester as much carbon. So by understanding the soil conditions, we can understand how to make the best and plant the best possible trees for restoration of these ecosystems. So my questions for my project are twofold. The first is, what is the biodiversity of the ecosystem? Like I mentioned, this place was clear cut. So the whole, one of the main goals is to reestablish the wildlife community because they were displaced in this effort. So it was really cool. I think this is probably the Bigfoot vision of the project, is we really want to see if we can understand this reestablishment, if we can track, deer and bear and different organisms coming back in the ecosystem by just taking EDNA samples. And then the second question is, what is the makeup of the soil microbial community? And this is to better help us understand insight into the health of the soil. So here is the forest where we work in. It's pretty big. And so when I came on, they just had forest management treatments. And so basically have an active and control. One where there is no roads in there and they're allowing it just to free form as old growth. And then one that does have roads. And then there's also patches of old growth that was still in the forest. And then there is patches of clear cut that wasn't restored. And basically we took samples all where you see the little orange dots to get a different, to get a good variety. And within these plots, it's important to note that we took samples where they planted different types of trees. So there's all different types of trees, their ages, and we took samples at all of those different plots. So yeah, here we are in the field, and I'll call out this photo because this is actually us. I'm in the field here, but this is a demo because you'll see this guy has a notepad. This is actually the reporter from the Seattle Times doing an interview, and then there's the photographer. So again, being at the Nature Conservancy, we do a lot of just outward facing things. So there are times where I'm doing research and out in the field, but I'm also doing an interview at the same time. So that's been something I've noticed a difference between, academia and here. But anyway, we took a whole bunch of samples, basically collected them in triplicate for just accuracy. We took both top and surface soil. And then we just also took some general vegetation and forestry data. Then in the labs, basically we have a bunch of dirt. Then we take it back into the lab, extract the DNA. We're using the Power Soil kit. Then we use polymerase chain reaction, or PCR, to amplify the DNA. And so I'm using 2 different primers right now for this project, for 16S for prokaryotes and 18S for eukaryotes. And so I have a video next. You have to bear with me because I show this video because I want you to see it's fun in the lab, and you have to have fun, but there's also a lot of lab work involved in doing this eDNA work. And so I guess the hardest, well, I won't say the hardest part, the longest part is the PCR process, because when you're using eDNA, you have to think about the primers that you're using, different things like that. So there could be chances that it doesn't work or it's not in your sample because, again, you're looking for whatever is there. And I should say that I'm using universal primers. Some people use specific primers because they know what it is they're looking for. But for us, we're using universal because, again, it's just, I'm trying to see whatever is there. So anyway, I'm showing this video of me in the lab after I had done a PCR, and now I'm going to run it on a gel and basically run this on a gel to be able to see if the primer that you tagged is present in your DNA. So enjoy. I'm Dr. Tiara Moore, and welcome to the lab. So today in the lab, we're going to be doing a process called gel electrophoresis. So I'll show you a sneak peek. We loaded our gel, it started to run, and we have to keep it covered because the stain that we use is light sensitive. But come here, look what we got. So you'll see that the gel, you'll see the DNA fragments, they are actually starting to run. So hopefully when we put this under UV light, we'll actually see those primers that we put onto our DNA fragments. Yay! All right, our gel is ready to run. So I just turn off the gel rig, unplug it from the machine. Oh my goodness, come look at this gel. So here are all the samples that we just ran, and we see that the DNA fragment moved, something happened. So hopefully we'll see our amplification. So I just take this whole rig, take it over to our imaging machine, which uses this UV light to get us a photo of our DNA. So I'm gonna move it around a little because we don't want any air bubbles. Close the imager. There's the air bubble. Make sure that it's on the screen, on my computer screen. Close the imager so you have one good one off because we don't want to have any of that nasty styrer on our imaging machine. And then run protocol. All right, guys, this is the moment of truth. I could have just been here for about 3 hours doing nothing. Wouldn't that be so sad? Here we go. Bam! Hey! So this says PCR worked, everyone. Oh gosh. excuse the Mrs. Rogers vibes, but again, when you are in the lab all the time, you do have to, spruce it up a little bit. But again, I just say that because it is important for primer selection in the eDNA work, and you really have to take some time picking out the primers that you're going to use and thinking about what it is that you're going to be doing and not taking yourself so serious if it doesn't work. All right, so after that process, after we run the PCR for all the different primers that we're going to use, we then clean them to make sure there's no excess proteins that we don't want, make sure it's the best, just the DNA that we need. We index them to be able to tell the samples apart. We quantify them to be able to know the DNA concentration, because then we pool the primers. So you'll notice in the video that was just, you saw maybe one bar. I think that one bar, and so that was one primer. But then in the end, when I send them for sequencing, I actually pull them. So down here, you'll see I ran another gel to make sure that both primers were present before we sent them to sequencing. So we'll go from 1 to the 2. So then we send them off to a magic lab sequencer where they find all of the genetic sequences that are in the different samples. And then they send us a huge data set back, and we run them through the lovely bioinformatics pipeline. We use an in-house pipeline that is available on GitHub. I have the steps listed, but basically what we do, we get the long sequence back, we trim off the primers, the indices, we merge the reads together, then we blast them against the database. We actually are curating a database right now to make it more polished versus the blast database. If you're familiar, it's a whole lot of stuff in there. Then we run it through Megan, and that's where you can get just a quick and dirty tree, and I have an example of that. We do a decontamination process, and then the data is then ready for data analysis. And so the postdoc who built the pipeline, Ramon Gallego, it is available on his GitHub if you are interested, and I may have the link later, but it is an open pipeline. Then after that, like I said, we run it through through Megan. And so Megan is this cool tool that lets you just quickly visualize your data. And so this is just an example of a screenshot of like me just looking at a part of my data of just running it through Megan. And so immediately you can just see the different species and different things. And so I just pulled out fungi real quick. And so you can just do a Google search and see exactly what that is in your different ecosystem. And so fortunately, because of COVID, I don't have all of the data that I would like to present because we got caught up in the lab. But I actually was able to get back in and submit my sequences. So I'm hoping to have the rest of the data for this project available. But the whole goal is for me to hopefully be able to understand if the wildlife community has been reestablished, if we can find their DNA in the ecosystem. I know there's going to be some questions about maybe history. And so for that, we did collect topsoil for these samples for this part of the project. And so basically what that means is that that was the most recent soil that was there. And so we're saying that that was the most, like that was the freshest DNA that could have been possible. For the soil health assessment, for the microbial community, we actually did a little bit of the surface at the surface soil. And so we got a bit deeper. And so we have those two different levels. And so again, we hope that we can see changes in or just signatures of the wildlife community, but most importantly, really see and understand the soil microbial community to really maybe get insight into the soil health. So we can, like I said, understand soil health to be able to make the best and the best possible conditions for trees. And so I put this photo back up here because again, just to bring this point home, understanding soil health, we can again have better more productive trees, but then also healthy soil is just better in general. When we look at carbon storage in the soil, there is 8,580 gigatons of carbon stored in the soil. And this is 2 times more than in the air and in vegetation. So healthy soil is the most important. So if I haven't convinced you of anything else during this talk, hopefully I've convinced you of this. Dirt is life, right? And by understanding soil conditions, by understanding biodiversity of the ecosystems, we can make more informed decisions on our restoration efforts. And so that is what I spend a lot of my time on, and what Maggie kind of alluded to, and I think I would be remiss to not talk about, is that I spend a lot of time thinking about biodiversity, but also diversity. And so I put these two articles up here. They came around on the same year. No progress on diversity in 40 years. Ethnic and racial diversity are extremely low among United States citizens and permanent residents who earn doctorates in earth, atmospheric, and ocean sciences. Worse, there has been little to no improvement over the past 4 decades. And this was written in 2018. I also wrote the article that had Maggie mentioned in 2018, the only Black person in the room. And I actually didn't even know about this other article about the no progress in diversity. This was me and my own personal experience as a Black marine scientist trying to thrive and trying to exist. And as Maggie mentioned, I was on a whole board of directors for a large science organization, but I still in the field wasn't respected or seen to belong. And so I recount these experiences in this op-ed that I wrote about how, when I introduced myself as a marine scientist, people ask me, "Oh, you can swim." Or when I would be at conferences, people would say, "Oh, do you know where the coffee is? Can you get us a refill?" And I was on the board of directors. So those are experiences that I felt that I was like, wow, what's going on here? What can I do to fix this? What can I do to cause change? Because this isn't right. This doesn't feel good. And so I thought my first step was, okay, well, I'm Black and I'm a woman. The intersectionality of that is crazy to deal with, because I never really know what's happening. Is this because I'm Black? Is it because I'm a woman? Is it because I look young and nobody expecting me to have a PhD? What is it? So I created and I founded a woke space. And a woke space is a space for women of color to simply exist. And we have these virtual meetups where we come together, we talk, we have paint nights and writing retreats, but we also have advocacy sessions, and we talk about ally support to really bring the group together to say, hey, we do belong in this space. We can have fun, even if in the labs that we are existing in, they don't look like us. We can create a space. And that has been awesome so far. We have blown up. We have other solutions that we're providing. We have merch. We write a blog. A lot of things that I heard a lot during the whole, I guess, racial uprising is that, "Oh, Tiara, we didn't know racism happened to you." I was like, "Oh, how about that?" Not only does it happen, some of you all contribute. And so we started writing this blog where we explicitly talk about the stories of women of color in the field and the things that they have experienced. So if you're interested, definitely check out a Woke Space blog. And if you are a woman of color and who wants to put a story out, we do provide an honorarium for that. So definitely reach out. And then we move towards actually ally training, because we've seen a lot of DEI workshops and things that are check boxes. To be honest, they're taught by older white men who don't have the experience of oppression. So we now use our stories to talk about the things that are going on in the workplace that are real time, things that happened to me, and say like how we could have been helped and how we could have made progress together. And so those workshops and trainings that we've been doing have also been really beneficial. So if you are interested in a woke space or finding out more about what we do, definitely check us out, and there's our information there. The thing about life, though, is that when you do stuff, more stuff comes. And so we founded a woke space actually at the before this whole COVID and everything happened. But then the pandemic happened. But then also we know 2020 was crazy. George Floyd was murdered. Breonna Taylor was murdered. And then weirdly, a Black birder was minding his business in the park, and a white woman called the cops on him and said he was harassing her when he was just trying to bird. And so, because Black people are resilient, a group of Black birders got together and created Black Birders Week. And it was the most amazing thing I had ever seen on social media. I went on Twitter one day and there was all these Black people outside with binoculars looking at birds. And I was like, what is this? What's happening? But it was really uprising to say, hey, we belong here. Stop calling the cops on us. You could get us killed. Did you not see? You see what's happening? You call the cops. And so from Black Birders Week, a lot of other STEM fields created, Black and Chem Week, Black Physics Week, all these different weeks. And so I was again on Twitter wondering, well, where's Black and Marine Science Week? And so I tweeted that and we created Black and Marine Science Week. And that was such a fulfilling week. I went from being the only Black person in the room to the only Black person that I saw maybe in seconds, because we did a roll call and we were introduced to hundreds of Black marine scientists. And it was a whole community building event. But then, again, because the week did go so well, but then shortly after the week, NSF released the data for PhD recipients for 2019. And so I look at this and I'm like, okay, let's see where we are. And I'm looking at the data for ocean and marine scientists from who graduated with a PhD in 2019. And I see the number is 4. 4 in the whole US, Black people who are graduating in marine science in 2000. This was in 2019. And I think what's funny, maybe why I tie to this so widely, is because I am one of them. So y'all know 25% of the Black people who graduated in 2019 with a PhD. So when we see this data, when we see the numbers, when we look at the other folks compared to our white counterparts of 212, you can see that the work still needs to be done. So we have decided to continue Black and Marine Science, actually, and we've created a YouTube series called BIMS Bites, and I'll share the trailer with you.
Welcome to BIMS TV.
I'm Dr. Camille Gaines.
I'm Leslie Townsville.
I am Alex Troutman.
I'm Dr. Jeanette Davis.
I'm Dr. Tiara Moore.
I'm Simone Barkley.
And this is BIMS Bites.
BIMS Bites is a weekly series featuring Black marine scientists presenting a small bite of marine science.
I'm biting into what is marine science.
I'll be biting into the water cycle.
I'll be biting into biodiversity.
I'll be biting into plastic pollution.
I'll be biting into climate change.
I will be biting into tidal marshes.
Make sure you tune in Fridays at 10 a.m. PST, 1 p.m. Eastern. We'll see you there.
And the whole goal behind the BIMS Bite series is to spread marine science in a way that we can communicate to the general public, but to also, of course, have Black scientists presenting their research that they're passionate about and they're dedicated to. We all know YouTube is a huge place. People watch YouTube videos for hours. So the goal is that we increase representation of the folks out in the public that just even see Black people as scientists. So no one would ever ask me again when I say I'm a marine scientist if I know how to swim. And it would just be known and seen that, oh yeah, we exist in this field. And so that's the goal of marine science, so of Black and Marine Science. So definitely subscribe to our YouTube. How many people? There's 52 folks here. So I should see 50 more subscribers today for Black and Marine Science. But we're really just trying to get it out there that we're here. We're trying to thrive and exist. And until people understand and see Black people in the field and know that it's a thing, then things aren't going to change. So in closing, my tips for students, for life: find the space you need to succeed. This road has been hard for me as a Black woman. I'll be honest. I used to hide it. No, it's hard. It's rough. White people do stuff. We have done some stuff. And it's like, but why? I just want to be a scientist. But I understood immediately that I needed to make the spaces that I need, or I was going to leave. Ask for help. Ask for help. Some folks may or may not help, but you have to ask. And there are people who will help. I think there is a negative stigma. I would say definitely from my community. Oh, we don't ask for help. We got it. No, you'll die. Ask for help. Network everywhere you go. You're always networking. I'm networking now. I hope I got people to subscribe to my channel. I hope I got people to sign up for a woke space. Every conversation you have, you should be talking about your research and the things that you're passionate about, because you never know who's listening. And then the last step is to never, ever, 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 ever give up. This field, just being in STEM and science, the work can be difficult and challenging when you put other things like racism and sexism and all these other things on it. It can be very challenging, but the goal for me is to save the world. That's the most important thing. And as long as I know that's my goal and what we can do, how we can help contribute and dismantle climate change, that is what keeps me up. So all the other stuff is secondary if I can figure it out. But I also know I even come from a place of privilege saying that, because so many other folks have just been wiped out of this field because of the attacks and the oppression. And I want that to stop. And I hope you'll help me. All righty, with that, I'll take any questions. Thank you so much for listening. Thank
So
you.
everyone, feel free to unmute yourself if you want to ask a question. Otherwise, you can write into chat as well. I have a question for you to start. Firstly, thank you so much for your talk. I really enjoyed it, and I really appreciate all of your outreach that you're doing. It's really amazing work. I'm interested in one of your questions relating to your eDNA work. Specifically, when you're doing that work, is it just a presence or absence of the organisms, or is there a way to actually assess like quantitatively how much of that organism is present in the whatever environment you're sampling?
Yeah, I think this is probably the number one eDNA question that we are working through. And so specifically quantitative data for what I'm doing, no. You can have relative abundance and more presence absence, definitely. And especially for this project here, we're really trying to see what can we find. It's very exploratory. I think it would be really cool, like the data that we see. So it's definitely a presence absence. But then, like I said, I guess for my other project, we can do relative abundance for like just how many like sequence reads are in a specific area. And so that's how you can see what's more abundant. But I wouldn't say, oh, there was 5 fish there or anything like that. No.
I have a question. I'm not on mute. No, I'm not. Thanks very much for your talk, Tiara. That was really interesting. I do a little bit of eDNA work as well. And one thing I was wondering about with the research you've done is how complete are your reference databases? Because that's certainly something that we've run into in terms of both false positives when we know what should be there, and we get hits that where the heck did that come from, which may be database error, but also, sequences that you just can't match to anything. What's your experience in the?
Yeah, I would say we definitely have that. I mean, those same, like there are some that are just like there's nothing, it's just reads, and there's some that's like, oh wow, this isn't in this place. But for the last project that I was working on, we did have a curated reference database that was made in the lab. And I probably actually I think that paper did just get published, so it's probably available. It's like a Crux database, and so we are trying to like build the best reference database. I think we're definitely running into that, and I think we're just like trying to compare to what BLAST has, but knowing that is just a lot of just stuff, and seeing like how can we make like the best potential database for like the location in the area. And I know that's what we were doing in the project.
So do you feel like you're getting close, or like do you have a long way to go before you get to that point?
No, I feel like maybe I shouldn't speak so freely because my whole group is in here, but I think we are good. Like I feel like we're getting better. I think that we're getting a lot more matches that make sense than not.
And so then if you go to a different geographical area, because I don't work with microbes, I don't know that how variable they are. Would you then have to start from scratch creating your database?
And so and that's actually what I will be that's exactly what I'll be trying next, because I'm in Washington now and I'm going to be using the California created database. And so hopefully, no, like I wouldn't, hopefully it would be the best, but I will be able to tell you.
Yeah, no, that'd be interesting to know that, I think. Yeah. How much transferability there is.
I see a hand, Sandra.
Hi, Tiara. Nice to see you in person. I just know Tiara from the Twitter sphere, but so yeah, very, very good talk. Thank you very much. It was very, very enlightening and very funny. Dirt is dirt. So my question, I think I have two questions more so, is again about your postdoc work. And I was wondering if the samples that you had were from different time points. So since the time they started restoring that place all the way till now. And then my second question was regarding, you discussed, the type of trees that you have and how that can influence carbon sequestration. I don't know how they went on this restoration effort, but do they, because if they want to sequester as much carbon as possible, do they go on with going with species that don't exist even in that region just to do that? Is that something done in restoration efforts?
Okay, great question. Thank you so much. And it's just so cool to be talking to you. So the first answer is no. So I came out, they like I said, they've been restoring this forest since about 2004. They heard about eDNA, is like, oh, can we do this? And so that was the first time when I went out during one time point and collected data. And so that's why I mentioned whenever we collected, I guess it was the freshest time, whatever had just walked around. That's what we're saying we got at that time point from the surface soil. And then for restoration, so they did put, different types of trees, different species. Now, as far as them being local, I just want to say, I don't know for the best answer. The type. So I mean, I know there's a whole lot of different trees and there's a different ages, because of course they didn't plant them all in one day. So it was a series. And so we have that data and that information as well that I'll be using as my metadata. So the year the tree was planted, the type of tree. They've also taken the just again, forestry management, regular data points. So the size of the tree they've measured each year, the tree cover, they've taken what is that called? What's satellite data? Yeah, satellite data over, so to understand the canopy. So all of that data I have over the years, but the eDNA is just a one capture.
Very much.
Thank you. I saw Maggie come off mute at one point, but I don't know. Oh, whoops, I see somebody else now.
Sorry, yeah, somebody else. Sorry, I'm not able to raise my hand on this thing. Sorry. I do have a question, but Tiara, first of all, thank you for hitting so many bases as you went along. As one of the older white guys who has benefited, we need a broader vision. So my wife's a teacher and I've been scribbling madly and I've given her stuff to pass on to her classes.
Yeah, we have a kid version of Bim's Bites too, so she can check it out.
That's on the list. And I've also forwarded your imposter blog statement to a former student of mine. But yeah, having science made up of old white guys hurts everybody. And you're encouraging everybody in the audience, the one thing to add to that, don't stop believing in yourself. Don't give up. You all need to believe in yourselves. You won't get through grad school without that. If you're worried you're not good enough, it's a good sign that you're thinking hard and working hard. You need to believe in yourselves, and we need more of you. Like Tiara said, there's giant challenges out there. We need everybody. So Tiara, thank you very much. I do have a real question. My phone rang partway through the call. When you were looking at the soil composition, did you see symbionts for any of the different tree species? Were there associations between trees and particular microbes?
So that is a question that we will be trying to answer. I did mention, so I got disrupted because of COVID. So I don't have all the data yet. So I haven't started digging into the current project data. But that's what I would like. I feel like that's what we're looking for. And that's what
Awesome.
we're trying to understand, like what goes where. Mm-hmm.
We have seen that in Ontario. There's some important forestry species. Like white pine was a climax species here. And for decades, they said, well, they're declining, but we can't plant them because they won't thrive. They needed a fungal symbiont. Once that was identified, now we are able to recover white pine forests. So, sorry?
Is this published yet?
This was published a couple of decades ago. This was before eDNA.
Okay.
But you're working in older landscapes. There's a better chance of symbiotic relationships between tree species and different microbes.
All right, Chris, I'm gonna have to look you up. Thank you. That's awesome. But yeah, that's exactly what we would love to see. Like, if I could find that, then I guess I'll be okay. Well, now you're telling me I'm just recreating what you already did, Chris. Come on.
I didn't do that.
Come on now.
Anyway, awesome talk. Thank you.
Thank you.
Tiara, I think I have the last word because people have to go at 1 o'clock, but you're going to stick around for just about 10 more minutes, right? But thank you so much. This was just wonderful. Wonderful to hear you speak and to see you again, although not in person, but just wonderful to see you again for sure. Maybe we'll reconnect at an ASLO meeting. I have one real question too.
Okay.
Because just because I started to dabble a little bit in the metagenomics in the stream with the microbes, and I don't feel like we have enough information to link functions or even diversity with ecosystem health and soil health. And so some of that might be, maybe this is a follow-up question to what Joanna was asking, but slightly different. So we do have a lot of work ahead of us, like which are the microbes that are associated with healthy soils? It's a little bit maybe a bioassessment. Like in streams, we use, if you have too many of the nematodes, your streams are no good, right? Or not healthy. So it's a little bit that type of information that I feel maybe we don't quite have yet.
Right. And so I think that's why we did the comparison with like having the treatments, but we also have samples from the old growth in the clear cut. And so I'm hoping that will allow us to give some information like, okay, well, this is what the old growth looked like, their community. And we saw these huge trees versus the clear cut and what that community. So have that baseline of what it is that we see comparing to the rest of the treatment. So that's why we have those in. So I think that to answer your questions, like, yeah, we do need more, but I think we're trying to do the best in this situation by using what was already there. Cause like I said, we do have that patch of old growth that we also have samples at. So we'll see.
All right, thanks everyone. Tiara will stick around for 10 more minutes, but those of you in the class, I guess, can go. That's some really good advice for the students.
Yes, and definitely, I mean, my email is there. So if you need anything, feel free to reach out. I'm always available, or hit me up on Twitter. That's my favorite place.
If you did have time for one more, I know there was one that came into the chat about how quickly does eDNA degrade, and could you look at deeper in sediment cores